In the early 1970s, the possibility of mapping whole genomes arose due to the prior discovery of bacterial enzymes that cut DNA at specific restriction sites and the development of recombinant DNA technologies and gene cloning. Nevertheless, one could not identify one single gene amongst thousands of fragments of DNA until Edwin Southern introduced his eponymous powerful DNA transfer and probing technique in 1975. He realized that restriction fragments can be separated on an agarose gel and then be transferred to nylon membrane by capillary action, the same way that blotting paper absorbs ink. Afterward, the blotted membrane can be incubated with a radioactive probe specific for the gene fragment of interest, which in turn becomes visible by placing an X-ray film on top of the membrane. This was called Southern blotting. In 1977, gene expression analysis ascended dramatically when George Stark and his colleagues replicated the configuration of Southern's transfer apparatus in an effort to transfer cellular RNA to chemically activated cellulose paper. This technique was named Northern blotting. 2 years later, an extension of this technique, making use of specific antibodies, was developed, and this faster and simpler approach for electroblotting proteins to nitrocellulose membranes is clearly preferred today. The name Western blot was first given to the technique by W. Neil Burnett in 1981. Prior to Western blotting, proteins are separated via polyacrylamide gel electrophoresis, or PAGE, by virtue of their molecular masses. A solution containing an anionic detergent, sodium didecyl sulfate, or SDS, which acts to both denature the proteins into their linear sequences and also to transfer negative charge along their mass, is added to samples. To ensure denaturation of the proteins, the samples are then boiled at 100 degrees C for 5 minutes. In PAGE, migration of proteins is dependent upon the percentage of acrylamide present in the gel. Samples are loaded into wells at the top of the gel. In order to ensure correct flow of current through the gel, running buffer is added to both compartments of the gel tank. Samples are removed from the heat and pipetted carefully into the wells at the top of the gel. Once all the samples have been loaded, the gel tank is connected to the current. In PAGE, smaller proteins will migrate faster and therefore further through the mesh-like constitution of the polyacrylamide gel. Larger proteins will travel more slowly, thus achieving separation of proteins by virtue of their molecular masses. Filter paper, pre-soaked in transfer buffer, is placed on the platinum anode before pre-soaked membrane, commonly nitrocellulose or PVDF, is added. The gel is then placed in direct contact with the membrane. It is essential no air bubbles are present between each layer of the membrane or gel sandwich. Before the addition of the cathode, a final layer of pre-soaked filter paper is added. The whole package is then clamped together. An electric current is applied so as to transfer the negatively charged proteins from the gel in the direction of the positive electrode. The membrane which is nearest the positive electrode effectively traps the proteins, making a carbon copy of the protein pattern seen on the original SDS page gel. In order to examine the efficiency of the transfer of the proteins from the gel to the membrane is usually stained for total protein. As it is important that the stain does not affect either the antibody binding or its detection, a protein stain should be chosen which might be easily removed, such as Ponzo Red. After incubation for about 5 minutes, the membrane is repeatedly washed in double distilled water until well-defined protein bands can be seen. The membranes used in Western blotting have a very high affinity for proteins. To avoid non-specific binding of the detecting antibody, which is of course a protein, to the free sites on the membrane, a blocking solution is used. Antibody is chosen which binds to a specific region of the protein of interest or a specific epitope on a group of proteins. The antibody is diluted to a recommended dilution in wash solution, which can also have detergent or blocking solution added to increase the signal-to-noise ratio. The antibody is incubated with the membrane for 2 to 16 hours, depending on the amount of protein to be detected and the affinity of the protein for its target. It is important to wash the membrane between each step to remove unbound reagents and therefore to increase the signal-to-noise ratio. Most primary antibodies used for Western blotting are not detectable directly, and therefore secondary antibodies, which are complex to an enzyme, are usually used in the detection process. The choice of secondary antibody will be determined by the species of animal in which the primary antibody was raised. The other consideration in the choice of secondary antibody will be the detection system, which is to be used to visualize the protein of interest, and this will determine the enzyme or tag which is complexed with the secondary antibody. Enzymatic detection is a commonly chosen option for Western blotting, and the most commonly used enzyme for this is horseradish peroxidase. Horseradish peroxidase is then detected using chemiluminescence detection substrates. The signal from these substrates is transient. Its sensitivity, coupled with the ability of this signal to be captured on X-ray film or digital imaging equipment, this makes it a popular choice for detection. Once the chemiluminescence signal has been transferred to the X-ray film, this can be then developed and the signal visualized.